Sotola. Yes.
Okay, welcome everyone. We'll get rolling here. I'm Fred Henson, your co-moderator. Patrick's here assisting me there with the camera. And our next speaker is Alex Sotola. We'll be talking about population genetics. Take it away, Alex.
Thank you. How are y'all doing? Yeah, 2 modeling talks and genetics talk. You guys are in for a treat. Okay, so yeah, I'm going to be talking about population genetics of wild and hatchery brook, brown, and rainbow trout in a river in Massachusetts. So, also, don't know what happened to all my slides here. So, a quick question that I want to start out with you guys to keep in the back of your mind, right? And that is, what is the ultimate goal of stocking or population supplementation? Right? It largely depends on the agency, the species, the situation, right? But I think they all fall under 2 broad categories. That is, enhancing a fishery, right? Increasing the number of fish available to be caught, or conservation focused, right? Helping struggling populations in the hopes that they can recover or rebound, all right? And I just want you to keep this in mind as we talk today. So, using genetic tools, assessing the genetic structure is a great way to assess kind of the success of those 2 goals, depending on what you're looking at. And there are 2 major outcomes, or broad outcomes, I think, that can happen when we have population or stocking of fishes. And that is, hatchery and wild kind of reproduce at random, right? It can be a lot, it can be a little, right? It can be completely random, different degrees. Alternatively, those hatchery individuals do not or are unable to really reproduce at all. And so using genetics, genetic structure, population genetic approaches, we can understand which of these is likely happening. So typically, when we do population genetics, especially of trout and some monids, we usually see very strong geographic genetic structure over small and large geographic regions, right? So here's a study on brook trout from Nova Scotia. They covered the entire, I have a laser, use it, Alex. They covered the entire peninsula, I guess. If you're from Canada and it's not a peninsula, don't yell at me. They covered that entire region, and you can see each color denotes genetic ancestry, right? So we have very clear geographic genetic structure, okay? But what's really interesting is that in this population, they also do some stocking, okay? So the local population margery here, and there's also this margery hatchery. Sorry, I use my hands a lot. Anyways, you can see they belong to the same club. They have the same color, same genetic ancestry, right? And that is because there's an active brood stock, right? But all of these other locations are also stocked with that same strain. We don't really see any evidence of reproduction because of the colors that you see, right? Think back to elementary school. We like to look at colors in genetics. There are other hatcheries there too, which do not or are separate from those populations as well, which clearly indicates very strong genetic divergence between the hatchery and the wild fishes. All right? Except for that population where they use the broodstock. And so, this area has been stocked since the 1900s, right? So well over 100 years. And you can see, look at that bar graph, and even if you prefer the pie chart with the geography, the only population that had any sort of signature of reproduction between hatchery and wild individuals was that margery population up here. And then, I can't remember exactly where they are down here, it's one of these. But regardless, this blue color is a pure wild, right? So you can see majority of those bars are blue, indicating very limited reproduction between hatchery and wild fish. Right? So, naturally, we might wonder why. What's going on? And, well, it could be genetic in nature, right? What are the effects of living generation after generation in a hatchery? Well, oftentimes animals become susceptible to domestication selection or genetic drift, right? Genetic drift being random change in allele frequencies, super powerful in small populations. What are small populations? Hatchery populations, right? So, these populations can evolve, artificial evolution, to a hatchery environment, or their genetics can change because of genetic drift, okay? And it's thought that animals bred in captivity will evolve to live in captivity, okay? And there we have some data on this, right? So for example, in Chinook salmon, after 7 generations of living in a hatchery, they genetically diverge from salmon that still live locally in systems near those hatcheries, all right? In lake trout, whoops, there we go, due to living in a hatchery system, relative to their wild counterparts, there's evidence of decreased fitness, right? Less egg production, slower growth rates, less fertilization success, among other things, all right? Attributed to living in a hatchery environment. So, putting it all together, overall, most studies do find very little reproduction and thus strong genetic divergence between wild and hatchery populations. And recently, with the advent of next generation sequencing, whole genome sequencing, genome-wide sequencing, studies have found evidence of genetic signatures of domestication, selection, or genetic drift, where they're in select regions, very specific locations of their genome. All right? Which brings us to our study, where we wanted to, this is a small snippet of it, but we wanted to basically assess the genetic structure of wild and hatchery brook, brown, and rainbow trout in the Deerfield River in Massachusetts. And then we wanted to assess whether we can identify any signatures of selection or drift, genetic divergence, from the hatchery environment. So, quick methods. Tissue samples were provided from all three of those species, largely from the Deerfield River watershed chapter of Trout Unlimited, along with some USGS collaborators. Again, we have all three species from this one river, and I should also mention that they have been stocked for well over 100 years in this system, right? So this has been going on for a very, very long time. And here we are, just to put it in perspective. Deerfield, use the laser, Alex. That's the Deerfield River, kind of northwest Massachusetts. Here we are in Saratoga. Here's Albany. Okay? Now, one caveat here that also we should keep in mind is that we have kind of a limited geographic range, right? All of our sites from the river are within about 38 river kilometers, okay? So it's not a huge geographic spread. That's something that we would love to fix in the future, obviously, to kind of get a better picture of the genetic structure in these populations in this system. Okay? So we took a genome-wide sequencing approach where we generated SNPs, or single nucleotide polymorphisms, right? Which, here's your basic genetics class for the day, is a single change in a nucleotide, right? That all of these are the same, except for one single base change, a G to an A, whatever it might be. All right? And the beauty of this is that we get a lot of power, right? We get a lot of data points. It's covered up now, but we got roughly 70 to 80,000 SNPs per species, right? Depending on the species, it's different for each. And here you can also see our sample sizes. Wish we had more Brook and Brown, but you get what you get sometimes. And so, but we do have a really nice rainbow trout sample size. And from this, I generated and analyzed two data sets. One I'm going to call the genome-wide data set, right? So that consists of all SNPs for a given species, right? Again, roughly 70 to 80,000 SNPs. But I also generated ancestry informative markers, AIMs, where you find SNPs, or genetic locations, with allele frequency differences, all right? Basically what you're looking for is SNP1 in population 1 has an allele frequency of 0.9 or whatever. The opposite, SNP1 population 2 has an allele frequency of 0.1. Do the math. 0.8 difference, that's a high allele frequency difference between those two populations. Ergo, it's informative, it's useful. Okay? And out of our 70 to 80,000 SNPs for each species, we ended up with about 3,000 to 4,000 ancestry informative markers, depending on the species. So, ultimately, I wanted to take this approach because I thought it would be useful and interesting to understand whether or not the genome-wide genetic differences are large enough or big enough for us to differentiate hatchery and wild individuals, or do we need to use select regions of their genome to really piece apart or genetically identify a wild versus a hatchery individual? Okay. I was about to ask if that makes sense, like I'm teaching. So hopefully that makes sense. Okay. So, and this will shed light on their overall genetic divergence, right? Is it genome-wide, or is it accumulating in distinct locations in their genome? All right. So, that really did get messed up. All right. First, we wanted to ask, what is the genetic structure of these three species in our geographic region, right? Is it driven by geography or their capture origin, that being the hatchery or the wild environment, okay? So I'm going to show you, these are ancestry plots, right? The color, again, here denotes genetic ancestry. We ran this for each species. I'm going to show you the genome-wide data set here on the left, on your left. And then on the right, we'll pull up the ancestry informative marker data set. We run this with an arbitrary number of genetic populations, K, we have K2, we have K3. So 2 genetic populations, 3 genetic populations, right? So here we have brook trout, genome-wide data set, K2, no big split, right? Which isn't entirely surprising because they are the same species from the same area. We would expect them to not really have genome-wide differences at the K2 level, but interestingly enough, at the K3 level, we do see the wild individuals are mostly in this blue group, with the hatchery individuals mostly red with a little bit of blue left over. Okay? Bring in the AIM data set, it's a completely different story. We see a clear split between wild and hatchery individuals at the K2 level, right? That suggests very strong genetic divergence in select regions of their genome, and in fact, if you've been paying attention, it looks like we have a few that might be even recaptures, of recapturing those hatchery individuals in the wild. And then that genetic split is largely maintained here at K3, and no useful information is added with that third population. All right, brown trout. Interestingly enough, you'd see almost no genome-wide genetic divergence at K2 or 3. A little bit of a split here, right? In the wild, a little bit of red, a little blue, mostly blue over here. But again, when we bring in the ancestry informative data set, we see a pretty clear split at K3 to K2. No new useful information added at K3. Rainbow trout, we see similar to brook trout, no real split here, largely all blue. But then here at the K3, we see the wild individuals mostly orange ancestry, hatchery individuals mostly blue. AIM data set, similar type story, right? This is really kind of cool because we're seeing some consistency and differences between some of these species with the wild in the AIM data set and the hatchery splitting here at K2. Now what's interesting, again I was about to ask, but this orange cluster and the blue cluster, orange cluster, blue cluster here, 2 different strains. So in this system, in the Deerfield River, they stocked 2 different strains of rainbow trout, which is we picked up on genetically, right? So even hatchery strains, are diverging more so in select regions of their genome, but genome-wide, they are also starting to accumulate genetic differences. All right, so we see no evidence of geographic genetic structure, right? All of the genetic structure is between wild and hatchery individuals, and we would have found that, we would have seen it because this analysis makes no assumption about population membership, right? It's agnostic to where those individuals come from. We do see weak genome-wide genetic structure for rainbow and brook trout, not necessarily the brown trout, but for all 3 species, we see strong genetic structure with the ancestry informative, that AIM data set. Okay. So then we wanted to take a slightly different approach and ask, can we reassign individuals back to their capture origin, right, based solely on their genetic data? If we can not, that does suggest a lot of reproduction between hatchery and wild individuals. If we can, it suggests little or no reproduction depending on what we find. And based on results from previous slides, we predict that we will be able to do that. Okay, and so we did that using a DAPC, discriminant analysis of principal components, where you take individuals, you remove their identity, where they're from, hatchery, wild. You say, hey, any random individual, is it genetically a hatchery, genetically a wild, right? So all of these individuals were captured in the hatchery. These individuals were captured in the wild. The color corresponds to their posterior probability membership to a population, right? So we could see right off the bat, just to hopefully make this super clear, this red individual here that's caught in the wild, that's likely a hatchery fish that we recaptured, right? Because it was caught in the wild, but its posterior probability is to the hatchery population, right? So both using the genome-wide and the ancestry informative data set, we were able to do that. Of course, you can tell it's much less variable for both wild and hatchery individuals with that AIM data set. And this holds true for brown trout, which is even more stark and strong, right? They have almost, did I miss it? Okay, cool. Almost all individuals have posterior membership probability of near 1 to their population of origin, except for this one individual right there. Now for rainbow trout, we see more variability similar to the brook trout, where most individuals, at least definitely from the hatchery, have very high probability to being a hatchery fish, which makes sense. But again, we see some individuals here that were captured in the wild that have high probability of assignment back to the hatchery population. Ergo, they are likely hatchery fish that we recaptured in the wild. Okay. So we can reassign them back to their population of origin based on the genome-wide and the ancestry informative data set. Ancestry informative is better, as you might expect, right? But we are unable to do so to their geographic location. I'm not going to show it because of time, but the only population based on geography we could reassign back to their original population was the hatchery fish. All other fish from the wild were basically equal proportions of every collection location. Okay. All right. So, select regions of the genome. Select regions of the genome, right? We've been talking about that a lot. So now, let's address that. Do we see signatures of domestication selection or drift in hatchery populations relative to the wild population? So, what we did was we took our 70,000, 80,000 SNPs, we found regions of high genetic divergence between those populations, and here I have the numbers, those locations, along with the totals, so if you're really good at math, you can calculate a quick percentage, but it's a very, as we might expect, a very small percentage of genetic locations that have high divergence between those two populations. We then took that, we mapped it to their locations on their reference genome, because we have pretty good reference genomes for all three of these species, and we found genes within 10,000 bases of each of those SNPs. And again, roughly 800 to 1,100 genes were found near these SNPs. Okay. Step 3 in the process is you find their functional group, right? What do they actually functionally do? What are those genes responsible, or what are they involved? What process are they involved in, right? So, interesting, we found 151 functional groups, so we whittled down all these genes to 151 functional groups. 55 of those functional groups were shared across all 3 species, 39 were found in 2 species, and 57 were unique to a particular species, right? So again, we're seeing some consistency across all 3 species, and also some differences, okay? Now, again, there's 151 functional groups, so I'm not going to list them all out for you, otherwise we'd be here for another 40 minutes, but these are kind of the big ones that I picked up on perusing through the data, right? Response to stress, morphogenesis and development, processes involved in reproduction, germ cell development, response to external stimulus, and cellular pigmentation. Right? I think we can all agree that these make a lot of sense, that might diverge in hatchery populations relative to wild populations, right? So, this is just a snippet of the ones that we found. And how does this happen, right? Well, this results from a shift in allele frequencies, right? So, we have our ancestral population. One population is going to diverge one direction, one population might diverge the other one, and that could be to genetic drift or domestication selection, okay, in the hatchery environment. So, ultimately here, genome-wide, there's weak genetic structure, right? It's much weaker than at select regions of the genome, okay? So we found evidence for a lot of genetic divergence in your genes, which are seemingly related to hatchery rearing, right? We went from about 3,000–4,000 ancestry informative markers down to 800–900 regions of high genetic divergence, and those regions are near genes which are seemingly related to hatchery rearing, right? And we found consistency across species, some differences, right? But it's interesting, 3 different species of trout, we found that consistency of where they're genetically diverging, thus they're responding to the hatchery environment in a similar way, potentially, right? Unless it's all random due to drift, and then what are we doing here? But consistency is interesting, right? So, just to revisit our initial question here, what is the ultimate goal of stocking, right? If we just want to throw a bunch of fish in the water that are available to be caught, that's great. Awesome, we're successful at that, right? You throw 10,000 trout in the fish, in the river, they're going to be there, right? But if our goal is to help struggling population in the hopes to not one day to stock again, conservation focused, I think genetics is a great tool to better understand how we're doing with that, especially because if these hatchery fish are less fit, do we even want them to reproduce with the wild fish, right? Because gene flow, alleles moving from poor fitness to what should be high fitness, right? Could be problematic. So, anyways, thank you all so much. If I have time for questions, happy to do so. I'd like to thank you.
Time for one question.
do you have any proposals as to making changes to habitat or hatchery management practices to make the fish more likely to reproduce with one another?
Well, I think the easy answer is replenishing your broodstock, taking from local populations and replenishing the hatchery broodstock. Based on other literature and kind of my thinking, that seems to be the best route, is for hatcheries, managers to actively manage their broodstock.
Thank you.
Thank you. Thank you.